Hi, I'm Rita Horvath, Director of Chemical Pathology at New South Wales Health Pathology in Sydney, Australia. I will discuss with you the medical needs for standardized test results from laboratory measurement procedures. In my talk, I will cover some key concepts and definitions, why we need standardization of test results, what should be standardized, who should define and maintain standardization, and the main beneficiaries of standardized test results. The global clinical laboratory test market size was around $190 billion in 2020, and it is anticipated to reach nearly $300 billion by 2030. According to various global surveys, on average, 15 to 20 laboratory tests are performed for each person in developed countries. Laboratory tests support clinical decisions related to screening, assessment of health risk, diagnosis, differential diagnosis, selection of treatment, monitoring of health status or response to treatment, and prognosis. Laboratory tests can replace other tests used as a triage test to select patients for further investigations or as an add-on test to refine the diagnostic process. It has been estimated that approximately 95% of clinical pathways rely on patients having access to laboratory results. It is a long journey until a newly discovered potential biomarker becomes a clinically meaningful medical test. Manufacturers often release new biomarkers without completing the full test evaluation cycle described by our working group in the European Federation of Laboratory Medicine in 2014. This has been particularly prominent in emergency situations such as testing for SARS-CoV-2 virus in the current pandemic. Early release of tests happens in spite of knowing that analytical performance influences the clinical performance of a test, and both impact on the clinical and cost effectiveness of testing. Laboratory tests, if not used in the right patient population for answering the right clinical question, or if they are misinterpreted, could potentially cause harm to patients, lead to wrong medical decisions and actions, unnecessary further investigations, and costs to both patient and the healthcare system. Laboratory tests are clinically meaningful and add value only if they are used and interpreted correctly. Interpretation of laboratory results depends on comparison of results to reference limits, for example, for screening, diagnosis, differential diagnosis, or risk assessment. If the results are compared to decision limits, again for screening, diagnosis, risk, monitoring, and if they are compared to previous results, mostly for monitoring health status or response to treatment or following up prognosis. Test results need to be compared to keep patients safe, to avoid confusion, diagnostic error, and harm related to testing, to aid consistent interpretation of laboratory results and medical decision-making, to support appropriate use and interpretation of medical research findings, share reference intervals and decision limits, to develop and implement diagnostic guideline recommendations with method-independent decision limits, to combine data in medical databases, to allow patients and clinicians to move across healthcare institutions, to avoid retesting and that way to save money, to support analysis and synthesis of big data, to facilitate evidence-based research and clinical audit of laboratory information, and finally to avoid research waste and again this way to save money for the healthcare system. To provide optimal laboratory service and reliable results for safe medical decisions, pathology results should be comparable across the entire healthcare system. This can be achieved by standardization and harmonization of test results. This aim of both standardization and harmonization is to provide uniform, equivalent, and comparable results irrespective of what methodology or measurement system is used in different laboratories. The term standardization refers to achieving harmonization by metrological traceability of patients' results to higher order reference materials and/or reference measurement procedures. The term harmonization refers to any process that enables establishing equivalence of reported values among different end-user measurement procedures. Harmonization ensures traceability to a reference system agreed on by convention. Metrological traceability is the property of a measurement result whereby the result can be related to a reference through a documented, unbroken chain of calibrations, each contributing to the measurement uncertainty. So, standardization has full metrological traceability, while harmonization has partial metrological traceability. Traceability requires commutable calibration materials. Commutability means that values measured for a calibration material and for patient samples have the same relationship between two or more measurement procedures for the same measurand. So to summarize the key concepts and definitions of the first part of my talk, for patient safety and evidence-based best clinical practice, laboratory results should be comparable. Comparability can be achieved by standardization or harmonization of test results. For standardization or harmonization to work for the benefit of patients, laboratory methods need to be traceable. Traceability requires commutable calibration materials, and maintenance of traceability requires commutable quality control and quality assurance materials. So moving to the key question of my talk, why do we need standardization of test results? And why this hullabaloo about traceability and standardization? So let me illustrate this with a simple example from the food industry. Food traceability allows us to trace all the steps a food has taken from its origin through its transformation process until it ends up with the customer on our dinner table. It is essential for controlling the risks that can affect the food supply chain and the health of the consumer. The main objectives of food traceability are that it provides accurate information to the consumer, it guarantees the quality of the product, it works as a quality control system, and it tests and certifies the withdrawal of food from the market. So in our daily life and for preserving our well-being, it is important to know what's being put on our plate. In response, farm-to-fork traceability is a rapidly evolving technology to meet customer demands for transparency in the food industry. So, what is the state of the art of traceability? In my workplace, in recent days, we have been discussing problems with bilirubin measurement in babies with severe neonatal jaundice. This baby, 2 days after birth, was found to have a very high bilirubin in the concentration range of up to nearly 25 milligrams per deciliter, in other words, 420 micromoles per liter. This result was measured with method A1 in one of the community laboratories. The same sample was remeasured with method A2 that was from the same manufacturer, but on a very different platform, and the bilirubin was remeasured as 27 milligrams per deciliter, which was way out of the Bhuthani nomogram scale, which is used as a guide to help clinicians decide when to treat patients with high bilirubin concentration and when to administer phototherapy. So as a result of these very high results, phototherapy was immediately commenced on this baby, and the baby was transferred to the hospital, where 10 hours after the first sample, bilirubin was measured with method B, and the result came back as 17.5 milligrams per deciliter or 300 micromoles per liter. The clinician called the laboratory and questioned the result, as he found it too rapid of a drop compared to the baseline that was measured in the community laboratory at 24.5 milligrams per deciliter. So therefore, after the phototherapy, the same sample that was measured by method B was remeasured again with method A2 in the community laboratory, and it came back as 22.8 milligrams per deciliter. That is much higher than by method B. Worth noting that in this case, all bilirubin results were way above the phototherapy threshold, and thus these discrepant results would not have changed the management of the patient. Had similar biases between methods been observed when results are at the border of the high risk and high intermediate risk zone, patient would have been monitored quite differently according to guideline recommendations. Another manufacturer recalibrated its total serum bilirubin assay in 2012. After calibration, the serum bilirubin decreased by 1.25 milligrams per deciliter, 21 micromoles, which led to a 39% relative reduction in the number of cases with a bilirubin above 15 milligrams per deciliter, or 250 micromoles, and a 51% reduction in the number of those whose total serum bilirubin exceeded the phototherapy threshold. This case illustrates that recalibration impacted the clinical diagnosis and management of a large proportion of patients, and the analytical performance of the bilirubin test impacted its clinical performance. The impact of recalibration was also shown on the frequency of phototherapy and on phototherapy readmissions. The use of phototherapy in birth hospitalizations dropped 59%, and readmissions for phototherapy dropped 53% after recalibration. Again, this case clearly illustrates the impact of analytical performance on treatment decisions and clinical outcomes, that is, on the clinical effectiveness of testing. Recalibration has also affected cost effectiveness. It led to a significant reduction in use. For every 10,000 deliveries, recalibration resulted in a reduction of 1,300 patient days per year and 4,500 fewer bilirubin tests per year, with a total savings of 1.4 million for every 10,000 deliveries in just one US institution. This study provides a powerful example of how small changes in the analytical performance of methods can lead to large changes in diagnosis, that is, to clinical performance, and treatment decisions that led to differing clinical outcomes, in other words, to differing clinical effectiveness and the cost effectiveness of care. To improve the consistency of phototherapy for neonates, the standardization of bilirubin methods will still be required. At this point, it is worth mentioning that we do not know how the current manufacturer's bilirubin methods relate to the assay that was used to construct the widely used botany phototherapy nomogram. This highlights a common problem related to lack of standardization, that is that guideline developers rarely take into account and record what laboratory methods were used to derive decision limits, which then survive often for decades and well beyond the life cycle of certain old laboratory tests and technologies. So, to summarize this part, we need standardization of test results to avoid diagnostic and therapeutic errors, to avoid harm to patients, and because analytical performance of tests that play an important role in a clinical pathway impacts clinical performance, clinical effectiveness, and cost-effectiveness of the care pathway. This leads us to the next question: what should be standardized? We should prioritize for standardization tests that play an important role in the clinical pathway. These tests are the part of a definition of a disease, such as troponins, for example, for the universal definition of acute coronary syndrome, cholesterol, HDL cholesterol, LDL cholesterol for defining cardiovascular disease risk, creatinine, EGFR for staging chronic kidney disease and acute kidney injury, HbA1c for diagnosis of diabetes mellitus, glucose for, again, diagnosis of diabetes or gestational diabetes. And the other category of tests that require standardization covers tests that have well-defined, guideline-driven decision limits for clinical action. For example, HbA1c for monitoring diabetes, prothrombin time, INR for monitoring anticoagulant treatment, LDL cholesterol for monitoring lipid-lowering therapy, and drugs with narrow therapeutic window or target. A list of tests can be found on the International Consortium for Harmonization's website. This database has 143 entries by now, of which 63 has high, 60 medium, and 20 low medical impact. In terms of harmonization status, 71 is considered adequate, needing only maintenance, 28 active, 29 still requiring attention, and 10 incomplete, 3 inactive, and 2 unknown. Worth noting that total bilirubin has a reference method, but as demonstrated with our case scenario, further efforts are needed to maintain its harmonized status, and the performance of serum bilirubin assays probably need to be investigated with more care in neonatal sample matrices as well. Again, to wrap up this part of my talk, what should be standardized? So we need to standardize tests that play an important role in the clinical pathway and impact medical clinical decisions, tests that are part of the definition of a condition, and tests that have well-defined guideline-driven decision limits for clinical action. Moving to who should define and maintain standardization. To answer that question, I call upon again the food industry, which refers to the regulation of the European Parliament, and that requires that all participants in the supply chain are compliant with a food traceability system. This includes, of course, farmers, breeders, producers, transporters, everybody down to distribution. The regulation also requires that the state, including the relevant ministry, which must control the production and processing of food, ensures that all in the supply chain comply with the relevant European regulations. If an incident takes place, the state should ensure that the food security is guaranteed. So these principles are equally applicable to medical laboratories and how they should define and maintain the comparability of their test results. At this point, I would also like to highlight the ethical dimension of standardization. Quoting the authors of this very thoughtful paper from Belgium, standardization should be a first step in harmonization of the whole diagnostic value chain. The manufacturers also have ethical obligations, which can be one of two types. Oftentimes they use the contractual theory, whereby the vendor lives up to the claims about their products, or the due care approach, whereby they ensure that the consumers are not harmed by the test. So standardization is truly a social responsibility to contribute to the safety and well-being of patients and the improvement of the healthcare system. As such, diagnostic industry, regulatory and government authorities, standardization bodies, patients, and the medical community should collaborate in order to harmonize the testing process. We should be mindful, however, that standardization per se does not guarantee that any new test will be fit for purpose. A well-standardized measurand can still be harmful if it is used on the wrong patient or at the wrong time by the wrong user for the wrong question. Manufacturers should work with pathologists and clinicians to prove and refine the purpose and role of the new test in a clinical pathway and provide evidence on the clinical performance of the test in real life. This is now also required by the new EU IVD regulation and the FDA regulation, and raises the question when standardization is required or when it is at all feasible. So should standardization be done before any clinical studies are carried out, or should it be done later after there is more experience gathered with the use of the new test? So what should we do with old tests also that have been long in use without standardization or harmonization? These questions I purposefully leave open for the discussion in the forthcoming session. Finally, let me elaborate a little bit on who benefits from standardized laboratory results. Going back to my favorite food industry example, the application of a food traceability system benefits all the stakeholders. The final consumer is correctly informed, companies are more productive, they register fewer failures in their processes, and finally, the health authorities are able to manage any incidents that might arise in a more efficient manner. Again, we must be aware that just having a better informed consumer about traceability is not going to fully protect the individual from harm. What will reduce harm is when the relevant health authority proactively assesses and mitigates the health risks, actions the incidents, and prevents that they happen again. So to wrap up the last part of my talk, who should define and maintain standardization, and who benefits from standardized test results? In my opinion, everyone who takes part in the provision and use of laboratory results, that is manufacturers, laboratory and clinical staff, professional organizations like JCT, LMI, FCC, the external quality assurance providers, guideline developers, and so on, regulators, policymakers, patients, society, and maybe we can discuss in the interactive session other players as well. So when standardization is required, I think this is again a question that would need to be discussed at the interactive session. Should it be done before or after a new test is released to the market? I'm sure there will be some heated debate about it. And finally, what should we do with all tests that are not standardized or not fit for purpose? So, to close, if the food industry can trace its tuna from ocean to plate, we should also be able to do better in the medical laboratories of the 21st century. So thank you very much for your attention.